Okay, let's get started. Hello, everyone. I just want to go ahead and start by thanking everyone that participated for the past two sessions. And I want to extend a warm welcome to those who are starting today on our journey to the world of bioinformatics. Welcome, everyone. So I just want to quickly go over a couple of instructions for the webinar. And I just want to tell you that your mic is muted. We've had over 700 or I think around 800 participants right now, which is very exciting. And since your mic is muted, you can only type in your questions. All the sessions are recorded. I know a couple of you have questions about you not getting the video session so far. The videos are being edited, and you are going to get in your email, you're going to get all the sessions, the video live sessions, and also the presentations. You should be receiving them hopefully by the end of the day today. And Larry is going to be presenting today, and please send your questions and Larry will be happy to answer them live. So I got a couple of questions from you guys from the previous sessions. I just want to quickly go over them and introduce Larry, and he's going to get started. So I was told that the brief introduction on sequencing basics and introduction to molecular biology would be very useful for some of us with zero background in biosciences. I really appreciate your feedback, and thank you so much for coming forward and letting me know your feedback. I would definitely take this into consideration for our next series that we would do. And I do want to let you guys know that bioinformatics is a vast field with a lot of people with knowledge from different fields come and do bioinformatics. Sometimes like a programmer who has no knowledge of molecular biology, it's very hard to sometimes understand the basics, and some of the stuff can go right over your head, and I totally understand that. And so in our actual certificate program, we are going to have an introduction to molecular biology course that you can take. And also we're going to have another prerequisite, which is genomics and next-gen DNA sequencing methods and applications. So you could definitely take those prerequisites and understand the concepts before taking Paul's course. So the basic concepts of the next-gen sequencing and instrumentation would be taught in that course, and Paul's course will focus more on the analysis that's been done. So please go ahead and send in more comments during this week session. I really appreciate you all taking the time to attend it. So your feedbacks are very valuable and we love it. Another question from the previous session was, why should I be interested in bioinformatics? Is this field for me? A couple of people had asked me about this. So I just want to let you guys know, I want to take the current scenario right now and talk for a minute or two. So let's talk about COVID-19 and the relationship to bioinformatics. It's a very scary time right now. We're all on a lockdown, and there are a lot of physicians, including my husband, who's on the front line and treating patients, and it's very scary. And there's one thought that's running through my mind, and I'm sure the same thought is running through all of your minds as well, which is, when is the drug going to be discovered? When is the vaccine going to be available? So that's the question that we're all asking ourselves every day. And unfortunately, the vaccine and drugs take a minimum of 12 to 8 months. They need to go through their phase trials, clinical trials, and get approved. So it is going to take some time. And there are already more than 30,000 research papers that's been published on this topic just in the past 2 or 3 months. So you can see that every research group all over United States, all over the world, are dedicated themselves to publish papers and understand this virus and what's happening with it. So they've come to a conclusion of doing drug repurposing. I'm sure a lot of people know what drug repurposing means. I am going to discuss a paper really quick about what that means. So you basically take a drug that's already approved for other diseases and you repurpose them. So you kind of see how you could use the drugs that's already approved towards coronavirus. So we use a lot of docking in bioinformatics, which is simulating in the computer the interactions between the virus and the molecules that could be used to make vaccines and drugs. So I read this recently, a very interesting paper on network bioinformatic analysis that provides a lot of interesting take on how bioinformatics is applied towards COVID 2019. I really hope you guys will be able to read this paper as well. I just wanted to quickly summarize what it says. It says the novel network-based drug repurposing platform is to identify the drugs for the treatment of COVID-2019. So it's analyzed the genome sequence of SARS-CoV-2, and it's identified from the sequencing that SARS is the closest disease based on the genome similarity, and also followed by the MERS and the other human coronavirus diseases. So it used the AutoSeq pipeline using text mining and database searches, and it found out 34 COVID-2019 related genes. So taking those genes as seeds, they built a network and understood the drug prioritization algorithms, and they came up with 78 drugs to repurpose. So repurposing, as I said, means taking the drug that's already available and seeing what best suits our need currently. And with the manual filtering and based on the clinical knowledge, they came up with 30 potential repurposable drugs that could be used against COVID-2019. So some of these drugs you've already heard about and you've been seeing in the news, which is very exciting. And I think this is a great paper, and I hope you guys would read it. And it's very exciting. So coming back to the question of why you should be interested in bioinformatics, given the situation current in our world right now, I feel like everyone can contribute towards the human lives. And it's very important that no matter what we could do, it doesn't matter. You don't have to be on the front line manufacturing the drug or making the vaccine yourselves. But I think doing a small paper or just publishing something or just getting a small information can contribute towards the final goal of finding the vaccine and the drug. So I think bioinformatics plays a huge role in it, and you should definitely read some papers about it. And it's a very, very interesting field. So I hope I answered your question. So I want to introduce, I don't want to take up Larry's time. He has some exciting stuff to present today. He's going to go over statistical analysis and modeling for bioinformatics. I think he's going to focus on linear regression today. So he's a PhD candidate in biomedical informatics at Stanford University. His work is focused on developing statistical and machine learning methods for use on single cell epigenetic data and to understand the effects of this disease and aging on histone modifications. He has a bachelor's degree in bioinformatics and a biology from Loyola University. And he's extremely talented, and he's going to start teaching with us in early 2021. His course is Statistical Analysis and Modeling for Bioinformatics and Biomedical Applications. It's a core course, and we are extremely excited to have you, Larry. Do you want to take over?
Wonderful, thank you. All right, so we'll go ahead and get started. I'll share my slide deck today. All right, wonderful. So with this biostatistics workshop, I want to introduce the concept of linear regression, statistics, and kind of give you an intuition of when you're doing linear regression, how does it work, and what kind of problems I can apply it to. So let's go ahead and get started. So first off, we can ask the question, what is statistics and biostatistics? So if you look it up on Wikipedia, it's the discipline that concerns itself with the collection, organization, analysis, and interpretation of data. We are particularly interested in biostatistics, so we're interested in biological data. So the way I think of statistics is as a process. We want to understand something about the world. However, we can't measure everybody in the world. So for example, if we want to learn something about humans and medicine, we can't go out and measure every human. Instead, what we do is collect a subsample, and how we do so and in what manner is statistics, this collection process. Then we take and measure something about the subsample, which is a scientific measurement. How we measure it and what kind of strategy we have for measurement is also related to biostatistics and statistics. Then we find a model or some way to organize and interpret the data. Then at the end of it all, in a lot of cases, we try to apply what we learned about our subsample to the whole population, and that's statistics as well. So statistics can be broadly organized into two groups, descriptive and inferential statistics. So these have different goals. So descriptive statistics has the goal to describe a sample. So some examples of these are measures of centrality, which is the mean or the average, or like a median or a middle point, and measures of spread, which tell you how your data is distributed. So this is like a maximum minus a minimum or a standard deviation. And so this happens after you collect the sample and collect data from that sample. Then we have inferential statistics, and inferential statistics has a slightly different goal. which is to use the sample to describe and infer something about the population. Some examples of this include hypothesis testing, which we'll be touching on today, and regression analysis, which we will be focused on today. So some types of variables, just to make sure we have the same vocabulary for this workshop, let's describe what kinds of variables statistics deals with. First, we have categorical variables, of which there are two types, nominal and ordinal. So nominal, you can think of as category names that don't have an ordering. So for example, colors or species. You can't put them in any single order, and there are these categories. Then there are ordinal variables, which are similar to nominal. However, they have some sort of order. So for example, letter grades. A is greater than B, but these are still categories. Then we have quantitative variables, of which there are also two types. Discrete, which are countable integer values, so like population or counts of cases in the case of coronavirus. We can't have fractional numbers of these, so you can't have half a person. These are discrete values. And then continuous. And so these are variables that can take any value in a range. And these are often limited by the precision of measurement. So for example, height and weight is technically a continuous variable, but do we go around saying someone's 180.7586 pounds? Usually not. We usually round off to a certain number, but these are continuous values. So another classification of variables is very important, independent and dependent variables. And I will be using these terms a lot during this lecture. So independent variables are variables that you can measure in an experiment. They are your predictors. And by convention, they're located on your x-axis whenever you're plotting them. So an example could be fertilizer amount for a plant on your x-axis. Dependent variables, however, are variables you are trying to predict, or your predictee, and by convention, they're on your y-axis. So if I'm looking at fertilizer, I may look at fruit yield from a fruit tree on the y-axis. So now that we have some of the basic vocabulary, let's talk about a little bit about linear regression and its purpose and its goal. So the goal of linear regression is to predict a continuous, and this is very key, continuous, dependent variable from an independent variable or a set of independent variables. So an example might be to predict plant height from fertilizer amount, but we can't do something like predicting eye color from height using linear regression. That will not work. It is not a continuous variable. So linear regression take a common mathematical form, which is y equals beta 0, b0 in red, plus beta 1x. So beta 0, let's start with that, is the level of the dependent variable with no independent variable. So if you remember high school algebra or anything like that, this is similar to y equals mx plus b. This is your b term. Beta 1 is the change. As I gain one amount of independent variable, say 1 pound of fertilizer, how much does my dependent variable change? So you can tell a lot by looking especially at beta 1. So if beta 1 is equal to 0, you have a flat line and your independent variable does nothing to your dependent variable. So then your equation is just y equals beta 0. If it is positive, you gain dependent variable as you gain independent variable. So in our example of fertilizer and plants, as we add more fertilizer, we'll likely get more growth or more fruit produce. If it is negative, as your independent variable gets larger, your dependent variable will get smaller. And this, you can just tell this from the sign on beta 1. So I like to think of statistics using examples. So I'll introduce one now. So with coronavirus going on, let's just say we're a set of researchers trying to understand the length of time the coronavirus can survive on cardboard using fluorescence. For each of 6 separate experiments, we note the amount of time it took for all the fluorescence to disappear. So let's just say this technology doesn't actually exist that I know of. And this data is actually just a made up example, but we'll get to real data later. But we've done 6 experiments and we've treated cardboard. Let's just say we can make the virus glow, which is, we can do a lot with different types of cells. And we can measure how much time it takes for all that glowing to disappear. So in this case, in experiment 1, the fluorescence disappeared in 20 hours. In experiment 2, the fluorescence disappeared in 28 hours. Experiment 3, it took 24 hours to disappear. So we'll start with this kind of toy example to get kind of the basic ideas of linear regression down. So first of all, whenever we approach a problem, we have to think about the experiment and whether a regression model makes sense. So in this case, we are trying to predict the number of hours coronavirus will survive. when you think about it, should experiment number have something to do with that? Is that an independent variable? And not really. Experiment number is uninformative. You wouldn't expect the time something will survive to be reliant on experiment number. So we shouldn't be regressing on this variable. So the only variable I have left is time of hours. So if I gave you 6 samples and asked you to tell me what does the population look like, what would you pick? Likely you'd pick the average, and that's the best we can do in this case. So the best predictor of a single variable, a model, is its mean. And so in this case, for every one of these variables, since I have no other information, I will just predict that my line will just predict that it'll be 24 hours. And this plotted out looks something like this. So experiment 1 is at 20, and I'll predict 24. My experiment 2 is at 28, I'll predict 24. Experiment 3, I actually get it right. It's 24, I recorded 24 and I'm predicting 24, and so on and so forth. And then this estimate is just a line, y equals 24. So one thing about this estimate is that this estimate only correctly predicts one experiment. How do we quantify the error for the other experiments? So clearly we predict this point very well. However, these points are not predicted well. So how do we go about quantifying this error? And so the answer to that is something called residuals. So the residuals are the differences between observed values, so what we measured and what we know is true, and expected values, what our model is predicting. So for experiment 1, we observed that the coronavirus lasted 20 hours, but we predicted 24. So our residual would come out to -4. In the case of experiment 2, we saw that coronavirus lasted 28 hours. We predicted 24, and so our residual comes out to 4. And in the case of experiment 3, we saw 24 hours. We predicted 24 hours, so our residual is 0. So residuals are simply the difference between observed and expected by the model. So if I were to plot these out, I can plot this out in a nice graph and label these residuals. So residuals have some interesting properties. So if you look at this model, what we see is that the residuals be above the line. If you add them up, they equal 7. If the residuals below the line also equal 7, -4 plus -2 plus -1 would equal -7. If you add up all of the residuals, they equal 0. This is a really important property of residuals. Residuals should average and sum to 0 and be normally distributed. So let's think about why this is and why this property is so important. So let's just say that the sum of residuals is less than 0. That means we have a situation kind of like this. That means our model is generally over-predicting our data points, and it's ineffective. Then we can go to the situation where the sum of the residuals is greater than 0. So in that case, our model is generally under-predicting, and it's also ineffective. If I were to try to sum up the data using these models, I would say that this doesn't work. So when the sum of the residuals is equal to 0, this is the perfect balance. My residuals are equal to 0, and all these points are right on my line. So this is just right. So now let's talk about the residuals being normally distributed, the second point. So in blue here is the normal distribution. You can kind of read this as a bar plot or density plot. Most of our data, if you'll see, the residuals is around 0. So we have a lot of residuals around 0. And as you move away from 0, we see less and less of these residuals. And so we want them to look like this. So we have a lot of residuals around 0, say a lot of points right on the line, and then a few points that are off, and very few points that are very far off. That would be like in no man's land here. If they don't, and let's just say the second condition isn't satisfied, it may look something like this. So in this case, we have some residuals right around 0, so right on the line. And in other cases, we have residuals very far off in this line. So that means that the model correctly predicts for certain values, the ones that have residuals of around 0, but completely fails for others. And that might be something we want to investigate. Is my model valid? Is my model actually good for this data set? Should I stratify out my data and maybe have 2 models instead of 1? And that might be a conclusion that we have to make. Or potentially, do I need to change the type of modeling strategy I'm using? instead of just using linear regression. So residual analysis is a very, very powerful tool in informing linear regression. So now that we know a little bit about residuals, let's talk about another version of error. So another way to represent error is using squared residuals. So effectively what we do is we just take the residual that we calculated before and square it. So in this case, experiment 1 has a residual of -4, and then it has a squared residual of 16. Experiment 2 had a residual of 4. It now has a squared residual of 16. Experiment 3 didn't have any residual, no error. And now it has a residual, no error again. So you may be wondering, like, why the heck do we want to do this? There's two reasons why we want to do this. So first of all, if we square residuals, we're emphasizing the larger residuals. We're really giving more oomph and more influence to the points that are further away. So in this case, you know, the difference between 4 and -4 and -2 is 2, but the difference between the 16 and 4 is a lot bigger, 12. And so I don't want a lot of this error. And my line, if we're using these squared residuals, will better fit that line. This helps models ensure that they're not missing these values that are very far off and are still taking them into account. It also, in squaring them, it makes all of the squared residuals positive, the sum positive, which is useful because it allows us to add them. So before, if I added all of the regular residuals together, my sum should be 0. And that's not super informative. That's informative for us to know that we're not overestimating or underestimating, but it's not really telling us much about how good the model is. However, if I'm doing it in this case and adding all the residuals together, it's really going to give us a powerful tool called sum of squared errors, or SSE. So let's talk about this, this metric SSE a little bit more. In our case, we have an SSE of 46. So the simple goal of linear regression, really down to it, is to create a model that minimizes the sum of squared errors, or the SSE. So if the SSE is equal to 0, we have a perfect model with all points on the regression line. All of my residuals are going to be 0, and I don't have any error. This is the ideal case. The larger the SSE, the more variation we have from our model, and the worse the model fits. And so this simple goal of linear regression and using SSE to create a model is called least squares regression, or ordinary least squares. There are other types of models as well. So even more complicated machine learning models and models used for AI use SSE to kind of optimize and make sure that the model fits well. So let's move on to a different problem now, instead of this one-dimensional problem, and go to coronavirus data that has been coming out recently. So let's just say we're a set of researchers trying to find the doubling rate and predict the number of SARS-CoV-2 infections in New York City for the coming days. The data we have is below. So we have data from March 13th to March 30th, and the number of cases recorded on all of those days. So so whenever you approach a problem, you really want to think about the type of statistics you're going for. So my two goals are to find the doubling rate and predict the number of infections in the coming days. So the first prediction, the doubling rate, is more of a descriptive problem. We are trying to look at this data and figure out a rate, which is going to be related to the slope that we were talking about earlier and beta 1. The prediction of trying to predict how many cases are coming in the next days is more of an inferential problem. We are trying to figure out what's going to happen next. And that's very important for epidemiology and for figuring out how to react to the coronavirus. So first thing we're going to do with our data is transform it just a little bit. So we can't really plug in dates into a linear regression model very well. So instead, we're just going to translate them to simple numbers. So I will call March 13th, 2020 as day 1, March 14th as day 2, and so on and so forth for the data I have until March 30th. And so we just sequentially increase that. Next thing you want to do is anytime you have data, you want to plot it. You want to see what's going on, and especially when you're trying to do a regression task. So if I plot this data, as we'd expect, it is very exponential. This is kind of how pandemics work and epidemics work. Thing is, with exponential data, we really don't want to perform a linear regression on the specific kind of data. It's not linear. Why would you perform a linear regression on nonlinear data? You should not do that. However, not all is lost. Let's just say linear regression is our go-to tool. We can always transform this data. And in this case, we'll apply a log transformation to all of the case counts to get different numbers. And so what happened is that we just basically took the log base 2 of each number in the number of cases to create a new column, log 2 of cases. And so now log 2 of 137 is 7.09, and log base 2 of 38,000 is about 15 or so. What this does, though, is kind of shrinks our range and takes some of the curviness and craziness out of our data in this exponential and turns it into more of something that looks like a line. We still have a little bit of curve, but this is a lot more linear, and we could fit a line to this data, and it's much safer to regress on. And this is a common strategy used in a lot of different applications and in papers and things like that. If you have data that's has a curve to it, try applying a different type of transformation to it to make it more linear and make it more workable for linear regression. All right, so how do we actually fit a line? So this is our data now where we're trying to fit date to the number of the log of cases. So our goal, more mathematically, is to fit a line that looks like y equals beta 0 plus beta 1. Yeah, and so I have a question live, and please explain why you're using log 2 and not natural log. So log 2 has a fun little property so that when we're looking at doubling time, the log base 2 is directly translatable to doubling time. The natural log, which is 2.8 something, does not directly translate to doubling time. However, if you are working with biological data, a lot of people will just take the natural log. If you're working with other types of data, log base 10 works as well. You will still kind of see the same effect of removing the curve from the data and making it linear. But in this case, log base 2 has some really nice properties for us that helps answer our questions later on. Okie dokie, so back to it. So our goal is to fit a line that looks like y equals beta 0 plus beta 1x while minimizing the sum of squared errors. We want that sum of squared errors to be as low as possible. Turns out there's a handy formula for fitting this regression line in this case. So in the case that you have a simple linear regression with one independent variable and one dependent variable, this is the equation that you would use to find the slope. So let's talk about its parts a little bit. So first off, we have x bar. So anything with a little squiggle over it is pronounced as bar. So x bar is the mean of x, of all x values. So in this case, it would be somewhere in this range, kind of halfway through. Y bar is the mean of y. And then xi is the x value for point number I. And then yi is the y value for point number I. So we have 18 i's, so we'll have 18 of these values. So what is this equation doing? So what we're doing is figuring out how far away from the mean is for each x and how far away the y is from the mean for each y and multiplying them together. And then dividing by the deviation in the x and squaring that. So what is this doing? Let's just take a couple of examples real quick. If I had a point way up here, it would have a high deviation from the mean in x and a high deviation from the mean in y. And it would increase my slope. And so the line would kind of tilt upwards to make sure I'm somewhat fitting that point and I don't have a lot of error there. The same thing goes for if I have a point way down here. Then my slope will slope a little bit downwards. The sum will go down and make sure that the slope kind of fits that point. And so we're kind of playing a teeter-totter game with this SSE to make sure that we have the line that most optimizes the fit using beta 1 and finding the slope. So now that we have beta 1 and we can solve for it, how do we get beta 0? Beta 0 turns out to be very easy. We can just take the mean of the y values and subtract beta 1 times the mean of the x values. So lines have a fun little property where any simple linear regression line will have to go through the mean of x and the mean of y. And we can use that back to just rearrange the equation y equals beta 0 plus beta 1x to beta 0 equals y minus beta 1x. So these two equations will get you your line. And if I plot it, this is what it kind of comes out to. Notice how it's not a perfect relationship. This line, if we're thinking about the residuals and thinking about to what we learned about those, it doesn't do so well right here, where our line is constantly over predicting the known amount of cases. And then it correctly predicts basically spot on one case. And then it under predicts in this range. And then we're over predicting again too. So this is whenever you fit a line, it's important to look at the residuals and see what's going on. So now let's interpret the model and figure out what it means and relate it back to our problem. So I have the equation now. So y hat is equal to 0.498x plus 7.45. However, this isn't entirely true. Y in this case is the log number of cases. This doesn't tell me the actual number of cases, which we have to translate back into using the log. So the actual equation I have is log base 2 of y is equal to 498x plus 7.45. So how do we interpret this? So we can interpret it as log base 2 of y will go up by 0.49 for every increase in x. So as x goes up in one day, we're going to see an increase of 0.49 in the log base 2 of y. I don't know about you, but that doesn't make so much sense to me. We can even put it in terms of cases, and it still doesn't really make sense. 0.49 times day plus 7.45 is equal to log base 2 of cases. That's not incredibly intuitive. That's not incredibly helpful. So let's actually do a little bit of math and transforming to get this back into the form that we can interpret. So what we can do is exponentiate both sides. So logs have this fun property that you can take 2 to each side, raise the power, and we can turn this back into an equation that makes a little bit more sense. So in the end, we get y is equal to 2 to the basically 0.5x times 2 to the 7.45. So let's actually interpret this model now that it makes a little more sense. So our model starts with 176 cases. So at day 0, we're going to be a little overestimating at 176. And with every 2 days, which we get from 1 over 0.49, we see a doubling of cases. So every time 0.49x is equal to 1, we'll see a doubling of cases. And then 2, 3, 4, and so on and so forth. So the doubling time of this data in New York is that we're seeing from March 13th to March 30th, a doubling every 2 days in coronavirus cases, which is quite a scary rate. So before we were talking about the residuals and how the residuals aren't necessarily ideal in this scenario. And so what's a way that we can kind of summarize how good the residual fit is and how good the fit of the line to the data is? One of the ways to do so is using the R-squared value, which we have here, which is equal to 0.91. So what does this mean and what is R-squared? Let's go a little into that. So R-squared is known as the coefficient of determination. It tells us what percentage of the error of y is eliminated when we use our model versus just predicting the mean of y. So if we look at the charts, on the left, we're predicting just the mean of y. So we're just going to say every value is, okay, I have a few questions, so let's go ahead and answer those. So when would you recommend fitting your regression with a forced 0-0 intercept? So in this case, with this model, we're letting the intercept be free. If the 0-0 means something, in the case of the problem, you could fit the regression line with a forced 0-0 intercept. And then also another question, will the number of people tested versus the number of positive cases have an influence on the model of results? Yes, 100%. So our model is just looking at will inherit all these biases that we're seeing in the data. So for example, if we have an under testing of data, we're going to see that. And we're going to go on in a few more slides. You'll see kind of towards the end of the presentation, you'll see kind of potentially a model on a different set of data that's also coronavirus that might exhibit some of the testing biases that we're seeing, that we know are happening from the news. All right, so back to coefficient of determination. So R-squared tells us what percentage of the error in y is eliminated when we use the model versus just predicting the mean of y. So this has a certain sum of squared error, which we can say is SSE mean. And what we can do is we can just calculate this error and then calculate this error for the one in the model, and then divide by the mean error. So this creates a sort of percentage. So you can think of R squared as like a percentage better than the mean. So in this case, my model is explains 91% of the error more than just predicting the mean. And R squared can be from 0 to 1. So 0, our model explains no more error than the mean of Y. And 1, our model will explain all of the error in Y. We have a perfect model. And so a 0.91 is generally very good across fields, but your R squared may depend on your problem set. So like I work with a lot of single cell data. If I get a R squared of 0.6, I'm incredibly happy. Other people, like in psychology, if they get a R squared of 0.5, they're ecstatic. In engineering and in some problems, if you get an R squared of 0.6, that's absolutely terrible. So this really depends on the problem, but it's a good signal that your model is fitting or not fitting in looking at R squared. So now moving forward, we figured out doubling time. California issued that stay-at-home order much sooner than New York City. Is the doubling time in California significantly different than New York's? With what we just learned and adding a little bit of statistics, we can definitely answer this question. So we'll add a California column to our data set. And so we start on the 13th with actually more cases in California than New York, and then we end with many less, about 8,155, as opposed to 38,000 in New York City. So as I mentioned before, the first thing we should do when we get any data is plot. So as was before, and as we'd expect, and what we saw in the New York data, the curve, when we plot the coronavirus cases versus day, does not look linear. So we should do what we did last time and take the log base 2. And now it looks very, very linear when we take this log transformation. And so we can kind of play with this. Now, I will be showing you statistical output from a programming language called R. If you want a little bit more exposure to R, there will be a session on Friday. But it's a very powerful tool to be able to model your data and actually explore your data in a very rigorous way, learning R. So I can fit a model just by typing in one line of code in R. So my model is equal to a linear model of the log of cases explained by the day with California data. And that's all I need to do to get all of these statistics. It's really, really nice. So after fitting the model, I get this. We always want to examine what's going on with the residuals. In this case, I'm looking at the residuals here. The mean of the residuals is equal to 0. Which is exactly what we want. That's a very good sign. We do have some residuals here and a little bit of residual error here. But in a lot of places, our line perfectly fits with the data. However, if I plot the distribution of the residuals and their density, so we don't quite see a normal distribution like what we saw before in the previous slides. There's a little bump. It's likely caused by these few points here in the 5 to 10 range. But overall, these residuals look pretty good. And we can continue analyzing this model. Note that your residual distribution, if you're looking at and examining the residuals and making sure you don't have any craziness going on, when you have less points, you will see a little bit more of this kind of bumpiness in your distributions. As you gain more and more points, you should see this smooth out into more of a normal distribution. And I just got a question for the case counts is the total or the active cases. And so these case counts were taken from California's Department of Health and New York City's Department of Health, and what they were reporting as reported cases. So obviously there's some testing biases here. Some places aren't testing at the same frequencies or testing the same types of people. So these are reported cases. All right. And so now what I can do is kind of with R, take a deeper dive into the statistics and different kind of metrics I can use to determine if my linear regression is good and what's going on. So if I want to do a summary of the model, I get kind of 5 columns that pop out from R. So the first column is our variables. This is the intercept, which is the y-intercept. We talked about that before. And this is beta 0 for our model. In this case, our estimate for the intercept is 7.75, which is really similar to the number we had before. We had 7 point something as well. So we're starting with a similar amount of cases for the model. And then day would be the model, what we're regressing on, would be our X value and what we're interested in finding a slope for. So in this case, our estimate for day 0, day is our slope parameter. The second column contains the estimates for these values. So in this case, the estimate for the y-intercept is 7.75, and the estimate for the slope is 0.295. And we can interpret this in the same way that we did with the New York data. We start with about 216 cases, which is a little bit different than before. And with every 3.38 days this time, not 2, we see a doubling of cases. And there's some other statistics here that we will also, in this table that we'll also talk about. Yeah, and so I have a few questions. Someone asked if we have, where does my p-value on the graph come from? We will talk about that in a few slides. A resource for discrete statistics as well. So if you're thinking about taking the UCSC course that we're offering in the winter on statistics, then we will be talking a lot more about discrete statistics as well. There's all sorts of different statistical tests and other types of methods that you would use for discrete statistics, not as much for continuous statistics. And aren't you at risk of overfitting if you target such a perfect fit of known data for the model, trying to minimize the R-squared error, even the mean squared error? Yeah, so overfitting is kind of a more machine learning concept. And you are right. So in this case, what we're doing is really trying to strongly fit our data. So in some cases, we don't want to do that. If we want to have a prediction task and predict our data in the future, very strongly fitting our data may not be a good thing. But in this case, to teach as a teaching lecture, I do want to strongly fit our data and kind of get the points across of linear regression. But you are correct. And we can also use other types of error metrics and sum of squared errors and see how that affects our models as well. Okie dokie. So there's a couple other estimates here, and let's talk about them. So we talked about the estimate. We know that. We saw that before in New York data, and then we're seeing it in the California data. Let's talk about the standard error. So in the next column, we have the standard error. The standard error represents how precise our estimate of the parameter is. In this case, we see very small values for the standard error, and for both the intercept and the slope parameter. Generally, a low spread of residuals will create a really low standard error. So I don't see very much deviation from my line. So I would expect my error and like my estimate to be very low. Yeah, and so let's go on to the next column. So the next two columns are really related. So we'll talk about them together. So t-value and probability greater than absolute value of t. So the calculation of the t-value in R is really, really simple. You just take the estimate and you divide by the standard error. So the t-value follows a certain pattern. With a very high estimate and a very low error, we get a very high t-value, which kind of means that a variable is important and informative. So you can kind of think of this value as a importance to the classification, or the importance to the regression, or an importance to the y value I'm getting out. How does this t-value work though? What are we actually doing? So a t-value is used with a t-distribution to determine what is the probability or chance that my estimate that I'm getting here is actually 0. So right now I'm getting estimate parameters, but what is the probability that, that this may be 0? Say if I add a little bit of noise or add a little bit more data or some other situation like that. Okay. So this is the t-distribution. So it's similar to the normal distribution we were talking about before, but let's think about it in a slightly different way. So we have this curve and it contains, we can think of it as having all of our probability under it. 100% of everything that can happen is in this curve. So the amount of probability under the curve cut at different values tells us that the chance that our estimate is 0. So at t-value equal to 0, we have all of the probability under the curve. So the probability that our estimate is equal to 0 is basically 100%. And so notice that this will only happen when your estimate is equal to 0. If you take 0 and divide anything, you'll get 0. Then at t-value 1, we slowly get rid of some probabilities. So there's less of a chance. You can think of these bars as chance of having a zero parameter. And so now we're going to go down to a 44% chance that the estimate is equal to 0. Then at t-value equal to 2, we have a 20% chance that the estimate is actually 0. So in our model for the slope parameter for day, we have a t-value of 83. So the chance that the estimate value is equal to 0 is actually super duper tiny. And that's given in the second column, the p-value column, the probability greater than t. And that's 1.5 times 10 to the negative 22nd. So we're talking 0.00022 times 1. So we are very certain that day is related to log of cases and that that estimate isn't actually 0. So I have another question from one of the viewers. If there are more data points with a higher magnitude, doesn't SSE-based modeling bias the model towards reducing the error in higher magnitude data while possibly ignoring the lower magnitude data? And yes, I will 100% agree that it will. So in different types of tasks where you have a lot of high error, so what they're talking about is a situation where I have a line and I may have a large cluster of points somewhere up here far away from the line. And in that case, the SSE will bias that line so it fits that data. What you can do to address issues like that is change from an SSE-based model to something, to a different metric. So an MAE, mean absolute error, could work. Or what you can do is have a custom residual function where I'm going to make sure I fit this data very well, and I'm not going to care that much about this data or have a certain distribution there. So yeah, so this comes into play a lot with different styles of machine learning, and that can be a very, very big problem. And another follow-up question, do I need to reject the model if p is greater than 0.05? So I love the 0.05 question. And so it really depends on your field. So in some cases, a p-value of 0.05, that's the statistical golden number, doesn't mean anything. So in my case with single cell data, a p-value of 0.05 means I have no conclusion here. In other cases, a p-value of 0.05 means, oh, this is great, or a 0.1 is very great. So it's generally just like a proxy of what's the probability that something will happen by chance. And the lower the p-value, the better. You don't necessarily have to aim at that 0.05. There's a lot of issues with aiming at that number as well. And if you look online, I'm sure you can find a lot of tutorials of like people showing like why p-value isn't necessarily the best metric. But it's important to kind of get, it's important, very ingrained in statistics and biostatistics. That's the issue. All right, so let's talk about one other statistic that we can look at. So we have multiple R squared and adjusted R squared. And so we talked about R squared before. It's the same idea. Adjusted R squared just comes in handy when you have multiple predictor values. In this case, in my X values, I only have days. But if I had a bunch of different variables, temperature, humidity, state, et cetera, this adjusted R squared would be the R squared I want to look at. And there's a few reasons for that. It's because as you have more variables, you would expect your fit to be better. The last variable given by our summary is the F statistic. And it works like a T statistics, just with a different curve. So the F statistic and its p-value represents the probability that just predicting the mean as a model is better, kind of like our R squared. But it's different than the T statistic in that it summarizes the whole model instead of just one parameter at a time. And so it looks at things as a whole with both B0 and B1 instead of just looking at them separate. And so this is what that P is less than 2.2 times 10 to the negative 16 comes from. It's this F statistic. And so that means our model is significant. So is there any formula to calculate death percentage or number of deaths? We could easily apply this kind of strategy with the death numbers and things like that. I've just done it with the case numbers for this example. So now let's compare the New York City model and the California model. So I've plotted both here. New York is in red, California is in blue. So New York City had a slope of 0.498 in log form and a doubling time of 2.0 days. California had a slope of 0.295 and a doubling time of 3.39 days. So looking at this model and looking at the doubling time of California in New York, you could probably say they're different, but that's not enough for statisticians. What you want is a probability that, that it is different, or what's the probability that they aren't different. So to really prove significance, we need a p-value to say that these slopes are different. What's the probability that this would happen by chance? So let's go ahead and kind of use a more advanced strategy to kind of address this. So when we were building these models, we were describing the log of cases, log of y, as a function of day. So we have day and log of cases. And we were building a separate model for each state. So we were building one model for California and one model for New York. However, there's another way to do this. We can build a single model for both California and New York by including state as a variable in the model. Now we have one model instead of two. So we can do this in R very, very easily. And if you look at it, this gives us a lot of parameters. And so let's talk about these for a little bit. So first, we have the intercept and the day parameter as we had before. These both have similar estimates to what we were seeing before and are both super significant. So the conclusion still stands there. Next, we have the state variable. And as you notice, this isn't significant at all. The p-value is 0.46. So 0.0, if it's significant, you would expect it under a low p-value. This p-value is really high. So what does this tell us? This variable represents what we know about cases by just knowing what state they came from. And we cannot predict the number of cases just by knowing the state. So we have a lot of points here where, like, if I just knew these points and I didn't know what state they came from, I don't know, I can't figure out just from state how many values I'll have. And so we need to look at this as an interaction between days. So this last variable is days combined with states. So you can think of it this way. If I have both the days since March 12th, and the state where it came from, how accurately can I predict the log 2 of cases? How important is this variable? And this interaction, which is based on the last line, is significant, which means that the slope day changes based on the state. And this is a p-value associated with that, which means there is a significant difference between California and New York City's coronavirus rate of change. So this is a really simple example of a multiple linear regression where you have more than one x variable trying to compute a y variable. If you want to learn more about multiple linear regression, check out the biostats course in the winter, and we'll definitely be going into this a lot more. All right, and so the rate of change that we've come into is significantly different. So I have a question. Why use days since 312 and not days since first reported case? There's no problem with using days since first reported case. However, in the beginning, the day is a lot noisier, where you may have a day where you have 10, 15, 20 cases, and then a day you have a lot less, and it's a lot noisier. So as a cleaner example, I've taken days since 312. And another reason why I've taken days since 312 is because both in California and New York, I had about 100 cases. So if you're modeling and comparing these values, you'd want to start at a similar starting point instead of just first case. More like, oh, there's 100 cases, this is already spread, this is still spreading, sort of an idea. So that's more of a choice on my end than anything else. All right, cool. So last thing, so we found out the doubling rates. We found out the doubling rates for New York City and the doubling rates for California and the doubling times, and we found out that they're significantly different. So can we use this as a predictive model for the future of what's going to happen in the next days after the data? So one way you can extrapolate and you can use linear regression is by plugging in a new x value. So we had x values from 1 to 18, from the 13th to the 30th. Let's just say I want to do April 2nd. I could plug in a value of 3 here and figure out where my y would be. Problem is, this is usually a bad idea. We can't just plug in the next x value and extend our line and figure out how many cases I'll have in the future. So how bad of a bad idea it is really depends on your model in the case. So in the case of New York, so this is the data for early April. If I were to use my model to try to represent this data and I extended this line, I would be very much overestimating the amount of cases I'll have, which is great, which means that flattening the curve and other types of other types of strategies we're taking to combat coronavirus are working. In the case of California, this extrapolation wouldn't be as bad. If I extended this line, I would still be overestimating, but I wouldn't be overestimating by like factors of 2 that I would be in like New York. You have to be very careful predicting when your data falls outside the range of your original data. So the key thing here is try not to extrapolate unless you're very sure about it. Generally, linear regression is a good way to model within your data and not necessarily outside of your data. In a lot of biological examples, I would have the full range of data. So let's just say I was doing an experiment with say 18 pounds of fertilizer or something, and I was measuring tree growth of 2 different species. In this case, if I plugged in something like 12, I could definitely predict where I would be. So you have to be very, very careful and don't extrapolate. And that you see this done all the time in models and the news and things like that, which is bad. So another question, doesn't the covariance of multiple variables give a first order idea of the interrelationship of their data distribution? So yes, so if you look at how the variables vary together, it can give you some idea of how they work together as well. And that's kind of what we're trying to get at with the interaction term that we were looking at before, the cases, the state times the day. All right, and so and so just a further lesson, be careful predicting when your data falls outside the range of your original data, don't extrapolate. So this has been a little overview of linear regression, and hopefully you've learned a little bit and gotten some intuition for the method and how it works. And I'll open it up to any questions over the question and answer for now. Thank you.
Thanks, Larry. We have another couple of minutes, so if you have any questions, please feel free to send it type it down. So Larry will definitely answer them. It was amazing, Larry. Thank you so much, and you actually explained the concepts beautifully. So it was well organized, and it was the current topic, and we really enjoyed it.
Thank you guys for listening. Hopefully it was informative. I will also post the code from all of these examples and analyses I'm doing online. If anyone wants to play with coronavirus data and linear models, it'll be there. And the Google Drive that we have.
That'll be awesome.
Okay. Yeah, and so how comparable is the predictive value of COVID-19 cases with the current COVID-19 cases in New York? So the predicted, this is the whole extrapolation problem. Our model would not be able to predict this. So our model is more for New York, especially in California, it's a little better. Our model is definitely overestimating in the case of New York. It's a little bit more accurate for California. And that might be an influence due to testing, that California might not be testing at the same rate or growing their testing rate at the same rate New York is. So another question, under what conditions is extrapolation valid? That really depends on your domain and what you're doing. In time series data, you generally don't want to extrapolate. In some other cases where you may have an idea of what's going on, you may be able to more look in the future and extrapolate a little bit more. But generally, I would say it's generally a bad idea to extrapolate too far away from where your model is. Would it make sense to normalize per population or take into account population density? Yeah, and so if I had a multiple linear regression model, what I could do is take into account other variables such as population density or the amount of people in a certain location and have that as an informative feature for my model and see if that would change the fit of my line a little bit more. Yeah, can I show you how to determine the doubling time again from the regression? We can go to that equation. Unfortunately, there are a lot of animations. Yeah, and so too far. So this would be the equation. You can definitely see it on video as well. What is the method you typically follow to determine the variables that drive the trend? Let's say the coronavirus, so we can use these variables to mitigate the trend in a specific scientific way. So that one's a little bit more difficult. We can, in a linear model, you can interpret the coefficients, right? And so with the right kind of linear model and the right kind of loss function, what you can look at is what are my coefficients on each x variable, that each independent variable, and how important are they to my classification of y. And so that could be a way of interpreting what's going on in the model. And these are simple models. In this case, we can't do that. But in the case you have a lot of models, you would be able to do that. Okie dokie. And so the instructor mentioned the course coming. If you email us, we can send you the course and all the course information, and we will answer that. And what types of job positions use these skills? A lot of data science job positions, bioinformaticians use these skills a lot. Linear regression is a first line kind of model for any kind of regression task. And so these kind of skills are really critical to understanding data and how to interpret and analyze data. And another question, you transformed your data. Well, if you just fit a quadratic or cubic. In this case, a quadratic or cubic wouldn't necessarily be ideal because it will still grow exponentially. With an exponential, you kind of get this flattening towards the end. So a log is just a common technique used to transform the data, but we could fit a cubic or a quartic or a quadratic to the data. Generally, you don't want to fit large degree polynomials to small data sets though, because then you're going to be running into a problem of really being overfitting and specific, but it depends on your task. If I wanted to work on applying data modeling or machine learning or AI in bioinformatics or solving bioscience problems, how much domain info in biosciences is required? I know a lot of people that have a lot of biology information. I have a bachelor's degree in biology. I know other people that have very little and kind of pick it up as they go. So I think the range is incredibly immense and it depends on your day-to-day workflow. If you want to run sequencing experiments, actually work with a sequencer and actually work with raw sequencing data, you definitely need a decent amount of biological information and biological knowledge. If you want to just build models or work on more of the machine learning AI aspects and have more of a discretized problem, you need a little less of the biological knowledge. So how are you better able to predict that the California model will predict better than the New York model? So this is just by circumstantial fact. So I can go to that slide. So I can't predict, I wouldn't be able to predict on March 30th if the model would do better. I could have a little sign of that and that these residuals are a little crazier than this, the residuals here. I wouldn't know that. But looking back, hindsight's 2020. Now I know that the New York model would not perform well, whereas the California model would. How do you know which transformation to use to fit a model? A log transformation is a commonly used one. There are other ones, Box-Cox transformation. Generally, I try to use the simplest transformation that will do the job, because as you transform the data in crazy ways, you lose interpretability of your model and you're not able to figure out what's going on with the model. As evidenced before, just thinking about logs isn't as intuitive as thinking about regular numbers and so on and so forth. So as you apply different types of transformations, it becomes even more difficult to figure out what's really going on. Do you have to make sure your data is bell-shaped before you analyze it? The actual data does not have to be. The residual should be. So if it's not, then linear regression is probably not the modeling method for your task. How would you combine state and date data? I'm going to upload the code, and if you want to take a look in there, we can build a linear model with using state and date data using statistical software like R. Can we predict the New York and California models by the end of May or June? So if you look up, there's a group at University of Washington that uses even more complex models. This is very, very simple and just an introduction. Very complex models to kind of predict things as we go a lot further out. So yeah.
Thank you so much, Larry, for taking a current topic and making statistics so interesting and making us understand the basic concepts. So thank you so much, everyone, for attending. Stay safe. Good night. See you guys.